Okay, so let you guys get signed in for the Kahoot. We're going to do the chapter 21 Kahoot to review chapter 21, and then we will jump into the chapter 22 lecture. When I was grading the lab unknown reports, this is by far the most lenient I have ever been grading those lab unknown reports. And I had to be, because so many students didn't listen to the instructions that I gave them. I mean, a lot. Probably a majority of students didn't listen to at least one of the instructions in the assignment. First of all, I don't normally ever let people submit it early and then me give them advice on changes to make and then let them make the changes. This is the first time that I've done that, and I won't be doing that again. You guys need to have your shit together before you submit the assignment, okay? So from now on, that's gonna be that. You had the whole semester to come in and like ask me about that stuff, and so future students will have that same opportunity, and if they don't have their stuff together by the time the report is due, they should have been doing that from day one. Second, I had a lot of students who submitted urea as their test explanation. Does anybody see a problem with that? Yep, I specifically said it in bold, in giant letters, highlighted in yellow, do not use the urea test. And I mean, I'm talking about not just 2 people, I'm talking about at least 15 people chose to do urea test for explanation. Okay. No, it wasn't just them. They were some, yeah, but it was people from all of my different sections. And usually the people that did it, I let it pass because they were rewording it in their own words. And there were some problems with it. And so it's whatever, they made the effort. And so I let it slide. I also let it slide that so many people weren't following the binomial nomenclature rules. I know I said that we weren't able to italicize, but then I also had students who would be like, sorry, I couldn't italicize. I was like, clearly, I would rather that be the issue, that you weren't hearing me say that you couldn't italicize it, than so many people submitting it like all lowercase or like all these capital letters here where they're not supposed to be or whatever. Because in the assignment, it literally says to follow the rules of binomial nomenclature. It says it in the assignments. So that was frustrating for me. It was frustrating for me with the urea test. It's also frustrating for me that people thought it might be acceptable to just say in their like test explanations on that last page, which was the most work of the whole assignment, to say things like the urea test is used to identify if the organism can break down urea, and that'd be all. Are you kidding? Right. So I'm like, anyway, so there was a lot of that with grading that stuff that like we had it our lenient semester, but hopefully you don't have to take my class again next semester. And if you have people that are taking my class next semester, that's not going to fly. We're not going to have that happening again. You're welcome. They're welcome. The people in the future, hopefully you guys won't be you. If they have to take my class, that they're welcome to come to me and discuss that stuff with me while they're preparing for that stuff before it's due. But not relying on like that last day to like tell me what I'm supposed to put in this report, please, sort of a thing. So yeah, and we still had a lot of people with blanks on their forms and things like that. I mean, it is what it is, but it was very frustrating for me to have these discussions of, hey, maybe don't choose the sim test, because if you choose the sim test, you have to talk about the S and the I and the M. And how many people did I end up having just talk about motility in the sim test? Like a lot. You'd be surprised. Maybe you wouldn't. I don't know. But I specifically said, like, if you're going to talk about sim or TSIA or something like that, or litmus, you have to talk about the whole test, not just the motility, not just the indole, not just, and I ended up letting people do that too. Because honestly, some people's explanations of the motility stuff was better than what most people's explanations were of the catalase, which was the #1 picked test. Oh, it's an enzyme that breaks down the hydrogen peroxide into water and oxygen, and the bubbles is positive. And that's pretty much what people wrote. And I'm just like, okay, I mean, you're not wrong. But like, I had so many people doing it. Like if I started counting off points for like the effort put into that, like none of you would have gotten A's on it. So it's just like one of those things where it's just like, I had to be very lenient, and it is what it is. And this is my one grace that I'm giving you guys for that lack of the study guide. Because I know already students complain, well, if you know, with the SSOC was still helpful with the test. Y'all think the freaking professors don't know what that means? You think we don't know what that means? Yeah, I bet it was easier having the answers to the test. I bet it was. Get over it, y'all. This is why. I talked with Brooklyn Statton, who is in the nursing program. She's like in her 2nd year in nursing. And I was talking with her about it. And she came in with a class of 100 people. And so they used to be that you had to get a 76% on the tests to progress on. And so they changed the rule to 75, but it's not just that. It's not they no longer round. So if you get a 74.99, nope, there's no rounding. So yeah, 75, but there's no rounding. So it hasn't really changed that much because they used to round. Also, you can replace one of your exam grades with the HESI grade, the HESI score, if you actually benchmark on the HESI. So there's that too. So it's not actually that much easier. But anyways, so they're sure they lowered the test grade, whatever. But yeah, she started out with the largest class that ever came into nursing, which was 100 students, and only 40 of them made it on to the next step from the first semester. So they lowered the grade so 70 could go on, and now it's 35 are left. You are kidding yourself if you think that just by going through these classes, you get a nursing degree. You're kidding yourself. Yeah, we have the highest pass grade on the NCLEX for the people that make it that far. And I've said it before, we're talking about not even half of the people are making it that far. You think you're going to be one of the people that make it that far. I'm glad that you are that, hopeful for yourself, but you have to actually put in the effort to get that far. You think that people like Brooklyn who've made it that far, you think she's like just lazing about and like not studying or trying to find ways to cheese the system? No, she works really hard to learn. She says she can tell the difference between the students that have made the effort to understand their prereq classes and the ones that haven't. There are students that will come in there and be asking questions about basic things like they'll have questions on their test in the third semester, having questions on the tests about like somebody having a viral infection, and the students are prescribing antibiotics. And if you don't see the problem with that, you don't belong in my class. You might as well just take it again, because you're not going to do well in nursing if you didn't learn what you were supposed to in my class. And those students didn't. They used the study guide to make it through their tests. And like I have said before, and I'll state it one more time, there are students who are making 100% in my class, 100% on the exams, on all their assignments and everything, who aren't even making a D on their tests in nursing. And you think that they're the exception? Imagine how the students that were getting Bs are doing. So you think they're the exception? Pull your little heads out of your little booties right now, because that's not the exception. That's the rule. We've got so many people in nursing that are realizing, oh, they weren't kidding when they said we have to learn how to learn. They weren't kidding when they said that we're going to have critical thinking. Nursing exams aren't just some of the questions are critical thinking. They're all critical thinking. My class is a baby compared to what you're going to have in nursing school if you get in. And odds are this semester you're not going to, because so many people are having to retake the first semester. They're not accepting very many new students this semester. Like dead ass. I've already got people with A's in my classes not getting accepted. More than one. So if you haven't heard back from your nursing school application yet, how many of you have heard back yet? Just, you don't have to tell me what you got. How many of you heard back yet? Okay. How many of you applied or are still waiting to hear? Don't hold your breath. So, is anybody willing to share? Did anybody in here make it into the nursing program? Yep, there you go. Right, so it didn't used to be this way though. I want to be clear with you guys. If you don't know, if you haven't talked to people from previous semesters, it used to be pretty much like, oh, if you've got A's and B's, it doesn't matter if you haven't completed blah, 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 but you can still apply. Now they're telling people, if you haven't got all A's, if you haven't got a good grade already in your midterm grade for micro, if you haven't got like pretty much elite, the elite are making it. That's who's making it right now. And it used to be everybody pretty much made it. If you passed your classes, then you don't have any fees, and you made it. Not this semester. If you were banking on going to nursing school next semester, probably not, is what I'm going to say. Probably not. It's the nursing program. Yes, I don't know what's going on with like vet tech or anything else like that. I have no idea. But I know in nursing, I'm already having students that are all A and B students, more than one telling me that they didn't make it into the nursing program. And it's because of reasons like that. You haven't completed this class yet, but that didn't used to be the case. You used to be able to get into nursing without having to complete the class. You could take it. They'd let you take it in your first semester. They'd let you wait.
I don't have the they want me to take the lab. I don't do that in this class.
Right. Right, but it used to be they would let you take that. They'd say like, oh, you don't have that yet. But they'd let you take it the next, when you start nursing, yeah. They don't let that anymore, yeah. So if you,
For here this year, because I was moved here, and they told me as long as I was taking micros that I would be able to get in because I had all A's and B's in all my science classes.
Mhm.
And then I didn't get in.
This is what I'm saying. So people who are taking micro, even people who have A's and B's in all of their science classes. She's taking micro, she's not doing bad in this class. And they didn't accept her. Because they don't have enough spots. Because so many people failed the first semester that they're having to retake it. They don't have spots for new people coming in because the first semester people are failing. Why do you think they're failing? You think the program got harder? No. Because they didn't learn. And they didn't learn how to learn. Like I said, it's the same people, the same exact people that got 100s in my class that are having to retake it. So. Right, and that's what I'm saying, is like, that's probably the bigger problem, and I don't know how, I can't solve that, right, because that's like one of those things like,
No, that's.
high school, and for most of you guys, how many of you guys took at least some of your high school classes, or before you came here, some of prereq classes, like, whatever, Comp 1 or whatever BS? How many of you guys took that stuff online? How many of you guys had online stuff in high school or anything like that?
I know credits for.
So, gotta tell you, never took an online class in my life. Not one, and I don't recommend it. They're not worth it, because you have to do all the work to the learning. You don't get to discuss it with the professor, like in here. You can't ask the professor during the lecture, hey, so when rabies, so I knew this person that got bitten. You don't get to ask those questions, right? You have to email, you have to save them up, maybe email the professor, that's extra work. Right, some of them, yes.
Like
So I'm a very communicative professor. You have me as the in-person class, and I bet I will, bet money that you've never had. Right. That's what I'm saying. I bet you would have never had, and you probably never will have, another professor as communicative and working as hard to trying to help you guys have the resources to learn how to learn. If I could, I would take the info and put it into your brains and have you just know it. If I could, I would do it. But I wish. Maybe in the future that'll be a thing. I don't know. But right now it's on you to learn it, right? And so like, unfortunately, if we don't, that's why I try to put so much work into helping you guys learn how to learn. Because unfortunately, if you don't learn this before you get to nursing school, you're going to have to learn how to learn there on top of the classes being harder, the professors being less communicative, and every single question that you have on those tests being critical thinking, not just 5 of them in your test. And every answer will be right. It's just a matter of which one's more right. All right. So that's why I'm hard. That's, I guess, why I am quote unquote mean, which I hear about a lot. If you talk to my hybrid, right, I'm just trying to be real is what I'm trying to be. But some people don't like hearing the truth, and that just makes them upset. And she's being mean. She's telling us we're not going to pass. You can pass if you do the work. You can. I hope that you guys do it. But if you don't, it's not on me. That's what I'm saying. So, it's my hybrid class, y'all. Anyways, yeah, so we reviewed our lab stuff yesterday, and because we're going to do that today, right? We're going to go up to the lab after this and go do lab review. And so we did that, and I talked about like the things that are going to be on the test. We talked about the stains, we talked about all the stuff. Literally 2 hours after that review, one of my students who was there for the review emails me and says, so what's going to be on the test? And then I'm just like, why did I even do this? Yeah. So, and like I asked them in their class, so I'll ask you guys this too, right? So I'll say this to you. If a virus infects, like let's say you get the flu, right? We're gonna talk about the flu today, but let's say you get the flu. It's a virus and it infects your lung cells. It gets inside of your lung cells and it presents that inside antigen on a molecule to ask for help. Which molecule does that get presented on? Okay, but even if you hadn't been right, because it is MHC1, right? Even if you hadn't been right, when I said that to my hybrid class, it was this.
At least we gave try.
Nobody talks in that class. Nobody tries to have the discussion. And I say, okay, this is the problem that you guys think by watching the lectures, it's the same thing as going to the classes. And it's not. And so I had them hold up their hands. How many of you have ever attended any of my in-person lectures? 2 of them, right? Which is even surprising to me, even just that 2 of them did. But I happen to know the 2 that did. And so, if you know, then it was Brandy Prophet who makes a pretty good effort to try to learn the stuff through the online stuff, and Donna Melton. And so they had, and I'm just like, you guys think that it's the same thing watching the recorded lectures? Oh, because who is actually watching the recorded lectures, right? That's the thing. It's like you'll turn them on and you'll play them. Yeah, yeah. But what I mean is like for the hybrid class, that's all they have. And so like, do you think they're actually sitting there like engaged in, yes, these notes? Or do you think they're just playing on the TV, like, well, that's on while they're like watching a TV, or to just focus, to stay in, but it's different in the class because this is all you got going on. And we got the opportunity for you to engage with me and ask questions and everything. And you see the difference. And this is why I only offer one hybrid section a semester, not to mention just on my side of things. I only get paid 1 extra credit hour for teaching a whole hybrid section. And so it's not worth the money for me to teach a whole other class that I only get 1. No, it's the same. Yeah. So for me, but like they have to do, but they have all the same assignments that you guys have, and everything like that. Their only difference is that for you guys, you get extra credit just for being here and like having engaged when I do have the questions, but they have to take those questions and type them out and give the answers in order to get that same extra credit. So that's the only difference, but they have the same quizzes, they have the same due dates, they have the same projects and all that. And yeah, I mean, if you looked at their projects compared to like this class, so I mean, no pressure on my unit 4 people, okay? But this class for unit 3 was exceptional with your projects, right? I mean, we did the songs actually covered the content. We had people do games, we had all sorts of stuff that was more engaging. But like, if you look at my online people, it's like, I am talking about the flu virus today. The flu is an influenza. And I'm like, what? You don't know what influenza? Or are you just literally like looking at the words, like reading it, and like you just aren't even thinking about the words coming out? But that is what it is. All of them are that way. And so like, yeah, the amount of effort is like here with you guys. Theirs is like here. That's what it is. And that's what they expected their online class to be. They expected it to be easier. I don't know what they were thinking. Yeah, online classes are harder because it's on you to learn the material. And their grades reflect that, let me tell you. But like, and I warned them, and I told you guys the same thing. On the hybrid classes, they get about a letter grade lower. And they're definitely showing that. And they're unit 3, man. None of the sections did like amazing on unit 3. Nobody's happy with unit 3. But if we said that this class got like a 67 average on I don't know what it was. I think it was like 63 or something. But they got like a 53 average on their test. So when I'm telling you guys, if you think you're doing bad, go visit those hybrid students, man, and talk to them how it's going. Yeah. Yeah. They just watch these lectures. Yeah. And so they have the same opportunities to join in on weekly reviews. They have the same opportunities to email me, to set up Zoom meetings, to come to my office hours. It's just the lecture part is entirely online. The lab stuff is still in person. And they're not doing any worse or any better on the lab stuff. But I think that kind of speaks a lot to the value that you get from being in an in-person class, because you're, I don't say forced, right? But you have to be here and we're talking to each other. And like you guys ask questions during the lecture because you're thinking about the stuff while we're talking about it. And like a lot of, like some other students have said, like, come to this class, it's like, wow, what disease am I going to hear about that kills me today? But it's kind of like interesting stuff, at the very least. But like, even for them, it's like watching a video about this stuff versus like being in the classroom talking about this stuff. It's a completely different vibe.
Helps me.
Yeah.
Because like, there's questions I don't even know that I don't know the answer to, and then somebody will ask and be like, hey, I need that.
Yeah, and so that sort of discussion opens up the doors for like, oh yeah, this person's asking this question. If you think to yourself, and I've said it from the beginning, if you think to yourself that you have a question, I guarantee you that most of the other class probably had that same question. They just might have not thought about it first, or they might have been too shy to bring it up or whatever. So keep that in mind. You guys, we're going to talk in the lab. I want to get into the lecture stuff first, but we're going to talk in the lab about some tools to help you learn for the lecture exam and for the lab exam too. But I feel like it'll help mostly for the lecture stuff, especially because we have a little bit of picking up some pieces to do with units 1 and 2. So I'm going to show you my note-taking recommendation, and then we'll get into like the lab Kahoot review stuff and that. And I think the lab review will be pretty helpful with talking through all the different tests and everything and all that. So anyways, let's do the Kahoot for now. Everybody make sure you're still signed in. mean, if you are one of the people that wrote about urease or just wrote about the motility or whatever, yeah, this is just me saying about how I'm frustrated that there were people that did that. But if you got your credit for it, you got your credit for it, man. And so be happy for that and move on. But my bigger complaint about it wasn't necessarily that about my assignment, but it makes me worried that if I gave you guys, I posted it on Canvas or I said it in class or whatever it was, especially the things I posted on Canvas. But right, what happens if your patient chart says don't give the patient penicillin? You say, oh, I would listen to that. Would you? Because you didn't listen to my Canvas announcement. Oh, that affects somebody's life. Does this not? Does your understanding of these diseases not help you? Right. So that's my point. It makes me nervous about my next hospital trip. Not that I wasn't already, because we know there's nurses who know their stuff and there's nurses that don't. And you will see that whenever you get into the nursing program, if you're going into nursing, you will see that. And you'll have your rounds and your clinicals that you'll be doing, and you'll see some of the nurses that you are with don't know their stuff, and you might know better. Right, yes, yes. So everybody has. So let's not be that nurse, yeah? Let's be on top of our stuff. We all have bad days, but to prep for that. And so another thing that Brooklyn brought up too is like, keep in mind when you get into, like when you finally, you make it through the nursing program and you get your, get your degree and you get your certification, everything like that. Whose responsibility is it to stay on top of their knowledge and to protect your certification? Yours. Nobody else is going to have your back with that. Not your charge nurse, not the doctors, nobody. So you have to be responsible for yourself. Nobody else is going to do it for you. Let's get started with our chapter 21 Kahoot to review this stuff. And we'll get into our respiratory diseases in chapter 22. Alrighty. What does the normal biota of the cardiovascular lymph systems consist of? We had a question very similar to this with the nervous system stuff. This is not. I feel like my hands are like, I don't know. I don't know if it's the humidity. Sometimes the humidity makes me feel like, yeah, this doesn't work. Oh, like shot over my shoulder and on my shirt here. All right. All right, so none of these. So the cardiovascular and lymph systems, just like the nervous system, do not have a biota. So we consider them to be sterile when it comes to microbes. Let's move on. Which term refers to when bacteria are flourishing in the bloodstream? They're actually multiplying in the bloodstream. They're not just transiently present. One of these terms refers to transiently present bacteria, and one of these refers to when they're actually multiplying in the blood. All right, so this is septicemia. So bacteremia is whenever you have bacteria present in the bloodstream, but if they are actively multiplying and causing an infection within the bloodstream itself, then that is a different thing. That's now septicemia, because you're becoming septic. To be septic is to actually have the infection in the blood system. Bubonic, well, we're going to talk about bubonic in a little bit, probably. I have a feeling. All right, which diseases are associated with the infection with Yersinia pestis? All righty, so this is pneumonic plague, bubonic plague, and the Black Death. So the Black Death is the septicemic plague. It's whenever this bacteria actually starts multiplying in the bloodstream. All right, how is Yersinia pestis transmitted typically? Which of the following is ways that Yersinia pestis can be transmitted? All right, so this is going to be flea vectors. typically like this is a bacteria that is harbored within the rodent population. And then the fleas will like bite them and drink their blood, and then the fleas will then transmit it to the host, humans in this case, but it could be other animals, right? And so this is technically a zoonotic disease that's maintained in that rodent population. So flea vectors, you can have pneumonic plague being transmitted from person to person from airborne droplets. What is a bubo? All right, so whenever the lymph node and the lymphatic tissue of the lymph node actually becomes infected by an organism, and then it becomes inflamed as a result of being infected, that is a bubo. So a proper lymphadenitis, where you have like your lymph nodes swollen from like the flu or something like that, that's just your immune system multiplying in there that causes your lymphatic, structures to swell up and everything like that. But whenever those lymph nodes become infected, that's whenever we have the buboes. All right, true or false? Epstein-Barr virus is a herpes virus which causes infectious mononucleosis. Is all of that true, or is some of it false? This is true. Epstein-Barr virus is a herpes virus, and it does cause infectious mononucleosis. Great work. So what type of pathogen causes malaria? All right, so this is protozoa. So protozoa, are they eukaryotes or akaryotes? Eukaryotes. So they do have a nucleus, they do have cell structures and everything like that. So the only akaryotes that we know about are going to be the bacteria and the archaea. We're going to talk about one archaea in the next chapter, not in this one, it'll be in chapter 23. But there's really only one that we ever talk about in the course. Most of the other akaryotes that we talk about are going to be the bacteria. So everything else other than viruses. Viruses are not cellular. They are acellular, so they don't even have cells. So viruses are out of that discussion entirely. Which part?
Well, not like the specifics, but just like how they became, because if they're not made of cells, then like they're
So that's
little aliens.
right. So yes. So to go back in time and to look at like the evolution of life would probably answer that question, but we don't know because we can't do that.
Yeah.
So that's the problem with that. But yes, so we can think of viruses as just being something like it's like a very basic machinery just for replicating itself, more or less is all that viruses are. And they aren't cells that can live or divide by themselves. So, but yeah. And so there's some gray area there. Of course there is, because there are some bacteria, very few, but some bacteria that actually do require a host and live inside of the cells and require host machinery and stuff too. So there's some gray area there, but we're not going to discuss that in my class. We're just going to leave it. Bacteria, viruses, and then the eukaryotes. So what constitutes a hemorrhagic fever? Alrighty, so we definitely have to have a fever for it to be a hemorrhagic fever. So it does have a true fever involved. And then hemorrhagic fevers are they are involved with vascular fragility from the inflammatory response as well as from effects of the infection. And all of the hemorrhagic fevers that we talked about, all the hemorrhagic ones were virus caused. And so this is going to be dengue, yellow fever, and chikungunya, and Ebola, and Marburg, and Lassa fever. They're all viruses. And so what constitutes hemorrhagic fever? Can a hemorrhagic fever potentially be caused by a bacterial infection? I'm sure that there are some, like when we talk about Rocky Mountain spotted fever, if that leads to hemorrhage, then it becomes a hemorrhagic fever, but they're not characterized by the bacterial infections. So this is a very similar sort of a choice that you might have on something like your nursing exams. We're like, could there be hemorrhagic fevers that are caused by viruses? There might be, but are they characterized by that? No. So our correct answers here would be causes fever and leads to vascular fragility that leads to the hemorrhaging out. Most of those are caused by virus infection. All right, which vector transmits the etiological agent, the causative agent, which causes Chagas disease? How is Chagas disease transmitted? Literally on Thursday night, I saw one of these on my like backyard door. Yeah, they're all over the place. There are, it's just you haven't really been looking for them. Yeah. So these are the kissing bug, also known as the triatamine bug. All right, last question. Which enzyme works to make a DNA copy of HIV's RNA genome? So this enzyme is called reverse transcriptase. There are 3 enzymes that are unique to HIV. That would be reverse transcriptase, integrase, and protease. So reverse transcriptase turns that RNA into DNA. Integrase helps put it into your DNA. It integrates it into your DNA. And then protease is an enzyme that, like, basically when HIV makes its proteins that make up its capsid and its reverse transcriptase and its integrase, all those things that get packaged into the virus, it makes it all in one long strand, and then protease cuts it up into the individual proteins. So those are all excellent targets for drugs for HIV. Great job, everybody. You did better than my Monday, Wednesday class. All right. All righty. So let's get into the lecture for chapter 22. We're going to be talking about our respiratory diseases. Yes, week 15. Somehow we've made it this far. All right. So respiratory tract diseases, we have the upper and the lower respiratory, basically defined by the trachea and the bronchioles and alveoli, the lung part versus like the throat and up part. So that's the upper respiratory tract. It's going to be your mouth, your nose, your throat, which is known as the pharynx. The sinus is the epiglottis, and the larynx. The larynx is your vocal cord box area. So we have anatomical defenses like your nasal hairs. The epithelium, or the lining basically of your lungs and your trachea and stuff, have what we call the ciliary escalator. So we have cilia up there that like help migrate up debris and mucus and things like that, constantly trying to move things out of the lungs so that it can't stay there long enough to cause infection. We also have mucus that helps like trap microbes. We have sneezing, we have swallowing, all to help just expel the stuff out of that respiratory system. The second and third line defenses are going to be your complement action. Levels of cytokines and antimicrobial peptides will be higher here. We actually have a higher immune response in your respiratory system than we do pretty much anywhere else, because of it being such a big site of microbes potentially entering the body through breathing them in. So, and that works to the advantage of these tissues. We also have secretory IgA, just to remind you guys of all those classes of antibodies that we have. That IgA is the one that's found in the mucus, whereas IgG, which is our workhorse one, is helpful for memory. The IgG is found in your bloodstream, not in your mucus. And so if we want protection in the mucus from antibody, then it would be IgA. We have a lot of microbes that live in the lungs. So there's actually quite a lot. It seems like we have a lot of bacteria. It's going to be bacteria by far the most. Then we would have our yeasts, which are a type of fungi. Then we might have things like protozoa and viruses and other things that might play a part in the biome as well. But it's a pretty high number of microbes that are part of the biome of your respiratory tract. The only thing that's going to, be more than that is going to be your GI system, which we already know there's a lot of microbes in your gut. But yeah. So the normal biota of your respiratory tract has a huge part to play in microbial antagonism. And in fact, a lot of people can actually harbor these, what we would consider pretty significant pathogens as part of their normal biome. But they're going to be existing in small numbers and maybe in like the sinus cavity as opposed to in your lungs where they might cause pneumonia or something like that. So some of these pathogens that can be part of people's biome will be Streptococcus pyogenes, Haemophilus influenzae, Streptococcus pneumoniae, Neisseria meningitidis, and those three there in the middle, Haemophilus, Streptococcus, Neisseria, those guys all were implicated in causing meningitis. Streptococcus pyogenes, we learned a little bit about how it can be implicated with impetigo, as well as necrotizing fasciitis, and then we're going to talk about strep throat stuff today. And then Staph aureus, which is more commonly associated with skin infections, but it can lead to other infections as well. And so people can just harbor these naturally in their biome, and these can lead to disease in different systems of the body whenever they get out of the place they belong or they overgrow. So let's start with the common cold. We also call this rhinitis. So we have inflammation in the nose that can also lead to inflammation within the sinus cavities and the throat and things like that. Many people have several colds per year. It's a 40 billion per year industry dealing with treating the symptoms of colds. It's defined by signs and symptoms, which are going to be sneezing, maybe scratchy throat, runny nose. You're generally not going to have a fever, but if you do have a fever, children might. It'll be usually a low grade fever involved in a cold. It's not going to be defined by that overall muscle achiness and fatigue and higher fever like we see with influenza. And so that's going to be the difference there. So what causes the common cold? Well, there's 200 different viruses that can cause the common cold. This would include things like rhinovirus, coronaviruses, not including COVID or SARS, right? But different coronaviruses, adenoviruses, and RSV can all cause cold symptoms, common cold symptoms. Now, whenever we see RSV presenting in infants, then they're going to have a much more serious presentation of that disease. But otherwise, people with otherwise healthy immune systems, like adults, they're going to have a cold-like signs, symptoms associated with it. It is transmitted by the air. Moving on to sinusitis. Now we're talking about inflammation in the sinuses. This could be caused by allergy or infection. And then we have there's 4 pairs of sinuses in your skull, and so any of these can become inflamed from infection or from allergy. You usually have sinus pain, congestion, pressure, headache, and even toothache from the pressure that's on the nerves associated with your teeth. The discharge is always going to be opaque. Typically, once we have sinusitis, it can be yellow or green in color as well. Almost all sinusitis is caused by viruses. So by far, most of it is viral. And so when you have a sinus infection and they're prescribing antibiotics, that's not actually usually treating the cause for most people. It can be caused by bacteria or fungi as well, but most sinusitis is caused by viruses. Okay, moving on to ear infections. This is acute otitis media. This is usually a common side effect of having a cold. We have the inflammation in our nose, potentially in our sinuses. Those tissues become swollen, and then we don't have the ability to clear out the pressure or equalize the fluid and pressure in your ears from the eustachian tubes that are basically running almost from your throat in your sinus cavities up into the ear. That helps with equalizing that pressure. So fluid will build up in the eustachian tubes as well as in the ear cavity. And then that will allow the bacteria that are just naturally in that area to overpopulate because we're not maintaining that environment as well. And this will be a feeling of fullness in the ear, loss of hearing potentially. Younger children might be irritable, have trouble sleeping, eating, or hearing as well. And it can cause the eardrum to burst, which is why we will install the tubes to help with the draining so that your eardrums don't burst. And that's the worst problem. You don't want to have to deal with that. So it sucks to have tubes in your ears, but it's better that than having your eardrum burst from the infection, which can lead to more complications and potentially permanent hearing loss, things like that. It's more likely to have that happen. And with the tubes, yes. Mm-hmm. Because the water will literally get into the ear. Because normally your eardrum is a barrier from the outside, but having tubes in your ears, the water can now get into the actual ear instead of not just the ear canal. I'm talking about in the inner parts of your ear. And so normally your eardrum is a barrier for that, but installing the tubes, it literally creates an opening. So that's to let the inside of your ear drain out any fluid, pus and things like that associated with the ear infections, but stuff can get in that way too. So that's the problem with that. Most of the time with ear infections, this is what we call watchful waiting. We'll wait and see what happens for a few days. And if the patient, if the kid usually isn't getting better, then we might treat it. It's usually associated with Streptococcus pneumoniae bacteria. And some more cases have been popping up being caused by Candida auris, which is a type of yeast. And this is a very difficult to treat yeast. It's more resistant to a lot of antifungal medications. This is not a communicable disease. You can't get ear infections from somebody. It's usually going to be an opportunistic infection caused from having a cold or something like that, leading to the inflammation in that eustachian tube. So that's this tube that will lead down into that throat opening area for equalizing that pressure. So yeah. I'm moving on to pharyngitis. Your pharynx is your throat. So this is inflammation of the throat. It can be caused by a lot of things, viruses and bacteria. Even the viruses that cause the common cold can lead to sore throat. Most of the time, if you have a viral pharyngitis, it's going to be less severe than the bacterial ones. Bacterial ones are more painful. They often have a fever, even lead to headache and nausea associated with this. But regardless, what we usually see with pharyngitis will have white pockets on the throat, white patches, and then difficulty swallowing, soreness, overall painful, and then foul breath associated with it. Bacterial cases are most commonly going to be caused by Streptococcus pyogenes and Fusobacterium necrophorum. So here's an image of pharyngitis. You see the pockets that are developing on the tonsils. This affects the throat tissue as well, not just the tonsils. So just because you had your tonsils out doesn't mean that you aren't susceptible to strep throat. You still get strep throat. You still have throat there. All right. So Streptococcus pyogenes is gram-positive coccus. It grows in chains. It's a facultative aerobe, whatever. So it has these capsules that it makes that helps it evade the immune system. And it also has molecules on its surface that mimics the human host cells. And so it's able to evade the immune system that way as well. If you have strep infection, usually it's going to start with strep throat, and then it can move throughout other systems of the body. It can lead to scarlet fever, where we see presentation of the rash over the body, and rheumatic fever, where we have the body trying to attack the bacteria and instead causes a temporary autoimmune effect that will lead to inflammation of the heart, and that's rheumatic fever. And we have glomerulonephritis. We talked about glomerulonephritis with the acute post-streptococcal glomerulonephritis from chapter 17 and the type 3 hypersensitivities.
My niece has an autoimmune disease called NML, and I don't really know, but from what they told me, it started as strep.
Right. So most people, when they get strep, if we can clear the bacteria from your body, like the autoimmune effects of having strep will go away, but not everybody has that. Sometimes it will be persistent. And so I'm assuming that's probably what happened there. It's not as common, but it does happen. It just relates to how their immune system responds to stuff. Again, it's sort of random how your body reassorts its genes. So you might not have gotten a good reassortment here. All right, so virulence factors. Again, we mimic host proteins, and so that can cause autoimmunity as well as helping evade the immune system. And it has several enzymes and toxins that it can make that help it infect the host. Some of these toxins are actually got from lysogenic conversion. So there's a phage virus that infected strep. And strep got infected with that virus, and now because of that virus infecting it, the virus brought genes to strep, and now those genes code for the toxins, and that's lysogenic conversion. Fusobacterium necrophorum, another bacteria that can cause pharyngitis. This is a potentially dangerous bacteria. It causes about 15% of acute pharyngitis cases, so it's not uncommon. And it is associated with this gram-negative bacteria that causes peritonsillar abscesses. You're less likely to have severe effects of Fusobacterium necrophorum if you don't have tonsils. But if you have tonsils, it creates these pockets for the bacteria to multiply in, and that can create pretty serious infection pockets that can actually lead to likelihood of the spread of the disease through sepsis or into the nervous system. All right, moving on to whooping cough. Whooping cough is also known as pertussis. This is not necessarily an old disease. It's still a problem in little babies and infants. They haven't been able to get their vaccines yet to protect from pertussis. So if you know Tdap is the, tetanus, diphtheria, and pertussis vaccine. So we're not going to talk about diphtheria here, which is another bacterial throat disease caused by bacterium Corynebacterium diphtheriae. You're welcome for not having to spell that one. But that causes a pseudomembrane in the throat, basically from the mucus and the bacteria, and it blocks off breathing, so you can't breathe. But pertussis is caused by bacteria Bordetella pertussis. And so this bacteria, you might have heard of Bordetella, and if you're going into vet stuff, then you've probably heard of Bordetella, because Bordetella is associated with kennel cough. And so this Bordetella bacteria. But in humans, Bordetella pertussis is associated with whooping cough, similar sort of presentation of disease. It starts out with just a normal upper respiratory tract infection, and then it can become into that's called the catarrhal phase. The catarrhal phase is where you're just having respiratory effects, like what we consider typical respiratory disease. But once you start having the uncontrollable cough, that's the paroxysmal phase. And this cough can be so severe that you will have broken blood vessels in the eyes. It can cause vomiting, and there will be hemorrhages in the brain. This is not an effect of having the infection, but from the coughing that you're going to have from the infection. And so you're coughing so much, it causes the burst blood vessels in the brain and the eyes and things like that. Serious coughing. We call this the 100-day cough. If you've ever had, like, you breathed in something and you're like, and you're like having to breathe in like that, that's whooping cough. Whenever this is going to last for weeks, months of that constantly. And I don't mean like you're going to have periods of coughing like that. I mean, like, that's it for weeks. And so this is a horrible coughing disease to have, and is prevented by vaccines. The convalescent phase, you'll have so much damage done to the ciliated epithelium of your lungs, those cilia that help move everything out. They're going to be so damaged after having the paroxysmal phase that in the convalescent phase, even though your body may have cleared the bacteria, you're going to be super susceptible to secondary infections from other bacteria because you can't clear things out because of your lungs being damaged. So you have a little video of a little baby. This is way more common in little babies now because as the elderly aren't getting booster vaccines, they can have the bacteria and not necessarily have serious symptoms of the disease. They go and visit the little baby in the hospital, and then the baby gets exposed to the Bordetella pertussis bacteria. They haven't had their vaccines because they're still a little baby. And so it's way more common in babies after we have our thing here. And this, it doesn't let me skip the. Yes, it does.
But these are good, sorry.
All right. So you get this, right? So this is pretty much what it looks like. Yes. Right. Yes. So they can do that. And so there's antibiotics that we can use to treat it, to try to help clear the infection faster as well. But yes, we want to provide oxygen to patients that have this to try to help with breathing and that. Yes. Deal with the inflammation. So the problem is that the bacteria are actually causing so much. So the steroids can help with some with the inflammation, but the problem is that you're opening yourself up with steroids to like other infections. So yes, so temporary use of steroids is probably advised, but with babies are going to be more susceptible because we're talking about little babies here. So with adults, treating with steroids is acceptable. With babies, steroids can inhibit like our ability to grow. And so treating with steroids is not usually advised in little tiny newborn babies, but it might be in serious cases where we're not getting enough oxygen in the bloodstream as a result of this. especially when we're talking about something that causes you to cough so hard that you're going to burst brain vessels. But Bordetella pertussis is a gram-negative rod. It is an aerobe, strict aerobe, and it has several toxins that are associated with the cause of this disease. Again, serious coughing, we do have vaccination available that is very effective, and we usually need boosters in order to keep that immunity up. It says a booster is necessary after the age of 11 years, but that immunity protection can wane as we become elderly and our immune systems are betraying us, basically. All right, so moving on to RSV. This is a virus, it says in its name, RSV, respiratory syncytial virus, and it is characterized by the fact that it causes the cells to basically merge with one another and create syncytia, which is multinucleated cells that have merged together, which is a cytopathic effect of the viral infection. So this really only causes serious disease in young children, but it causes like cold-like symptoms in adults. And whenever we had COVID masking and we had social distancing, there's not a ton of evidence necessarily to support that masking and social distancing was preventing the spread of COVID like at a significant degree, but it was likely that it was preventing that. But there is evidence supporting that it decreased the spread of RSV and influenza. And so imagine having COVID and also having RSV and influenza. You can have all of them at once. And so this actually masking and social distancing led to a significant decrease in RSV and influenza and may or may not have helped with the spread of COVID, but at least we were preventing second infections on top of the COVID, right? If you guys were happy enough to have some time on your hands to want to go to covid.gov, which is our government-led site about information about COVID, I don't know what to say about it. If you have time to look at it, it's become a propaganda page for this government's agenda. So yes, so if you have time to look, I'm not saying all of it's wrong or bad, but I am saying there's quite a lot of talk about how masking wasn't effective and how whatever. So anyways, it's not what you would expect, but hey, has anything from this administration been the things we've been expecting? I can't say that it has been. All right, moving on. So RSV, RSV signs and symptoms, you'll have a fever, you'll have rhinitis, pharyngitis, and otitis. If you have more serious, so again, cold and like symptoms, and it might become more serious and affecting the lower respiratory tract, leading to issues with breathing. Transmission, this is a highly contagious airborne virus transmitted through droplets in the air, as well as through fomite contact. Again, a fomite is like something like this keyboard or whatever that if I have RSV, I could have transferred the virus to this, but the keyboard isn't infected. We might say it's contaminated, but the keyboard isn't infected. But it can become a means by which we are using as a vehicle to transmit the disease. That's a fomite. That was, chapter 14 stuff. But yes, there are antivirals available for treating that. Then we have influenza. Influenza is caused by the virus influenza virus. And it belongs to a family called the Orthomyxoviridae, which I won't necessarily test you on, but it's a viral infection. There are different versions of flu, A, B, or C. You don't hear about C anywhere near as much as you hear about A or B. And A is the most common by far. Annual flu seasons can turn deadly for people, especially if they have decreased immune function and they're more likely to have secondary infections or have progression of influenza into like pneumonia, for example. Many diseases are called the flu whenever they're not. Flu is caused by influenza virus. It's not a syndrome like a cold. It's not a syndrome like pneumonia. Influenza is caused by the flu virus, right? Influenza virus. Flu is caused by that. So influenza is highly mutating virus. It has a structure. Okay, we're going to talk about that in a minute. Let's talk about signs and symptoms first. So influenza, the disease that it causes with the flu, we have headache, chills, dry cough, body aches, fever, stuffy nose, and sore throat. So the issue with the flu and the extreme fatigue. The issue with the flu that makes it really different from colds is that we have a higher fever, we have the body aches, and we have extreme fatigue associated with it. We don't usually see, I'm not saying you don't feel like crap when you have a cold, but you don't feel like crap like you do when you have the flu. Having the flu really will run you down. So this can leave the infection from the respiratory issues with flu can lead to secondary infections with bacteria and stuff that can then cause pneumonia. Much more serious in people with a reduced immune function. This is the flu virus. And normally I'm not going to ask you guys, I won't have any pictures on the test or anything, but this guy has 8 gene segments. Instead of having one genome, kind of like we have multiple chromosomes, the flu kind of does too. So these are its gene segments that code for different things for the virus, and there's 8 of them. And this is actually kind of important. We're going to talk about in a minute. But then we also have our surface proteins, and our major, most important surface proteins for these guys are the hemagglutinin and the neuraminidase. Hemagglutinin is our binding molecule for the virus that allows it to bind to the receptors of the mucosa in the respiratory system. And then neuraminidase helps break down mucus. It helps assist in budding of new viruses out of the host cells, and also helps with host cell fusion, incorporating that virus membrane with the host cell membrane when it enters the cell. So hemagglutinin is for binding. Neuraminidase is basically helping out the viral infection process by breaking down mucus and helping get in or out of the cells. So mutations of the glycoproteins on the surface of influenza, whether it is H or N. This is where H and N come from like H1N1 or whatever. Okay, so hemagglutinin and the neuraminidase. That this virus is pretty highly susceptible to mutation just naturally. You have general gradual change of the amino acid composition from just random mutations that can have reduced ability of the body to fight off the flu. And so this is like what we see from typical seasonal changes with flu. So every season we come out with new flu vaccines for the new strains of flu, and they're usually new strains due to just random antigenic drift. But sometimes we can have antigenic shift. And so the difference here is antigenic drift is just like from natural mutations, just from the multiplying process and some errors being made. Antigenic shift is whenever those gene segments become reassorted in a new host. And I'll show you a picture to help display that in a minute. But that's like basically like influenza having a new strain of flu because mutated, it looks different to your body. But whenever we have antigenic shift, it's so new to you, it might as well be this whole other different kind of virus. And your body hasn't seen that. So when we have antigenic shift happen, whether from H1N1 to H1N5 or H1N1 to H5N1, or whatever it might be, when we have this major gene swapping going on, you have no memory of those new guys. So the new guys that are being made from antigenic shift, you might have a little memory of some parts of flu for antigenic drift. It might have changed it a little bit. So you'll get the flu, but it'll just be like normal flu. But in antigenic shift, nobody in the human population has ever seen this guy. You have no immune protection from it whatsoever. And so this is where we get those severe pandemic outbreaks, like what we see whenever we're talking about bird flu coming into the human population, or we're talking about the 1918 flu and stuff like that. So what happens is you might have a duck flu and a human flu, and they infect a pig, and the pig becomes infected with both, and then we have a new virus come out with a different kind of neuraminidase or a different kind of hemagglutinin. So in here we have human influenza with a duck hemagglutinin on it. And so this would be this is antigenic shift. And so the way that I remember the difference here is like, if I think of drift, antigenic drift, if you're just drifting in the ocean might push you around, or you might be stuck in one place, but you're at the will of the ocean. Yeah, you're just drifting. Antigenic shift. You can shift yourself in the ocean if you have a paddle, right? Be active, more big change in your movement. But another way that I remember it is when I was describing this image to students one semester, I said shit instead of shift. Mm-hmm. And then, I think of like pigs. Now it reminds me every time, because I think of pigs rolling in poop all the time. Pigs are poopy creatures. Okay. So this is where I remembered, oh yes, this is when I say antigenic shift, that I actually think of poopy pigs and how this is gene reassortment from different species is happening in the pig host. And it's the most common reservoir for this sort of shifting to happen is the pig, but it reminds me of this image to think of it that way. So I don't know if it helps you or not. Okay, so you know how people tell you all the time, if it's cold outside, be careful, it's cold out, you don't want to catch the cold or whatever. And we think it's ridiculous because like the cold doesn't make you sick. Well, yeah, you're right. The cold doesn't make you sick. Like being cold outside, the weather being cold doesn't make you sick. But Cold weather is drier weather, and it dries out your respiratory tract. It makes you more susceptible to diseases. So you actually are more likely to get sick if you're out in the cold and the dry. So it's not untrue. You're not getting the infection from the cold weather, but you're getting the infection from the other viruses that you might have been able to fight off if you had your moisture and your mucus and everything like that. So cold air does make respiratory tract mucous membranes more brittle, and then that will facilitate with invasion of viruses. How do we prevent the flu? You know, social distancing and masking and hygiene practices is fine. And then we try to have vaccines available every year for the new seasonal flus. And then we're trying to develop a universal flu vaccine, but it hasn't been released yet. It hasn't been, you know, developed yet. So it hopefully will be available in the future. All right, tuberculosis. This is an ancient disease. Pretty much as long as people have existed, we've found people that have had tuberculosis. It used to be called consumption. And so that's the same disease. And so TB, tuberculosis is, you know, on the rise now because of the HIV epidemic, because we have drug resistant strains. Nearly a third of the world's population is infected with tuberculosis. That's something, man. Primary tuberculosis. It only takes about 10 bacteria to get into your body to be able to cause the disease on average. And these bacteria actually live inside of your alveolar macrophages. So they live inside of your phagocytes here. They're happy to live there. There ain't no phagolysosome going to break these guys down. They're happy to stay there. And so this leads to the formation of tubercles, which are granuloma tissue. So a granuloma is when your white blood cells try to wall off a site of infection. And so the white blood cells are literally trying to wall off that infectious site. And then as a result, oxygen isn't able to get to that area. And so the cells inside of that site will die off, become necrotic. And so those necrotic lesions become caseous, which is cheese-like. Yes. Okay. So beautiful. So these caseous lesions develop from the dead bacteria, from the dead white blood cells in the area, all from trying to wall off this infectious area. So those are tubercles. They eventually will dry out and become calcified after they've been in the lungs for a while. So you will actually have this going on in primary tuberculosis with very little to no symptoms whatsoever. And then people will usually go into a latent phase after that. So once the tubercles have formed and they become calcified, you can go on and live your life normally and not have any symptoms whatsoever. And then we will can inject people in the skin with tuberculin, which is a protein from Mycobacterium tuberculosis, and see if you have a reaction to it. If you do, this is the MANTOO test or the PPD skin test. If you do, it will become inflamed within 48 to 72 hours as a result of your CD8 cytotoxic T cell response. So this is your CD8 cytotoxic T cell response because these are inside bacteria. They live inside. And so you will have inside response into this. So it takes 48 to 72 hours because that is a delayed type cell-dependent reaction. Secondary or reactivation tuberculosis happens whenever those tubercles that formed and become calcified in the lungs, that those can become reactivated, usually due to decrease in the immune function, from stress, from all sorts of things like that. So you can actually have that bacteria dormant in the lungs for years. And then it will come back out and we will have violent coughing, greenish or bloody sputum, low-grade fever, anorexia, weight loss, fatigue, night sweats, and chest pain. This is consumption now. So consumption is always associated with secondary or reactivation tuberculosis. If it is untreated, it is a 60% mortality rate, which is why we hear so many stories about like in like the 1800s and 1700s of people just dying from consumption. So people used to try to treat this in, if you've ever heard of a sanatorium, it's not like sanitarium. It's not like insane asylums. It's not what I'm talking about. Sanatoriums were designed for sanitary purposes for treating tuberculosis. So they used to think that like, oh, we will just let these people get lots of fresh air and these nice like rural environments, and we'll let them sit outside and get sunshine and air, and they will get over the tuberculosis. But the tuberculosis bacteria is a strict aerobe. And so it likes the oxygen and is happy for the oxygen. And oxygen does not help with treating this bacterial infection. Now, what helps treating this bacterial infection is going to be antibiotics. All right, so this is caused by Mycobacterium tuberculosis. It is sometimes caused by Mycobacterium avium complex, which is a different serotypes of Mycobacterium avium, but that's really only going to be associated with AIDS patients. But most people will be getting the tuberculosis version of this. This is acid fast. We've already learned about that like a million times over. And we could do the acid fast stain on like sputum and things like that to identify this. If you are acid fast, you have mycolic acid wax on the surface of the cell. That makes it more resistant to things like drying out and cleaning with cleaning agents, things like that. So this is that acid fast test. The fuchsia colored reddish pink bacteria are the ones here. It is transmitted through fine droplets in the air. It seems to be more associated with close living conditions and is more associated with inadequate nutrition, low immune function, poor access to medical care, lung damage, and genetics, especially can make you more likely to have it. These are some testing methods like the PPD Manto skin test, immunoglobulin reactivity tests, which is like ELISA and things like that, PCR, obviously acid fast staining of sputum and chest x-rays. If you have tuberculosis, we can't really treat it whenever it is latent. It's harder to treat because it's hiding out and being protected by the body. But if you have active tuberculosis, we will treat you for 2 months. We will give you rifampin, isoniazid, ethambutol, and pyrazinamide. Whichever one works best over the first 2 months. We will continue to give treatment for 4 to 7 months until you have cleared the bacteria. 6 to 9 months of antibiotic. And some of us can't stick to 10 days of antibiotic treatment. And so it's no wonder that we have drug-resistant strains coming out now. Now we're moving on to pneumonia, which is a syndrome. Pneumonia is characterized by inflammation and fluid in the lungs. This is going to impede the ability of oxygen to get into the alveoli and provide oxygen to the bloodstream and get rid of the buildup of carbon dioxide. All right. So most of the bacteria that can live in the lungs to cause pneumonia are going to have to be able to evade phagocytosis or avoid being killed by the alveolar macrophages, which is the case with like tuberculosis. We have them living inside of the macrophages, like they're happy to be there. So it has to be something like that or be able to evade the phagocytes because we do have pretty strong immune function in the respiratory system. So viral pneumonia is usually tamer than bacterial pneumonia, but it can be caused by viruses, bacteria, or fungi. So we're going to talk about some of these guys. This is going to be characterized usually by upper respiratory symptoms first, and then we have progression into pneumonia in the lower respiratory tract, where we'll have chest pain, fever, cough, and discolored sputum, and the patients might look sickly due to a lack of oxygenation as well. Here we have the fluid buildup in the alveoli here, and the bacteria can actually like survive pretty well in that fluid and multiply within that fluid. That collects in the alveoli, and the alveoli aren't able to be exposed to oxygen, where we'll have the gas exchange occurring within the lungs to oxygenate the blood. The most common bacterial cause of pneumonia by far is going to be Streptococcus pneumoniae. It is community-acquired pneumonia. This is gram-positive cocci. It comes in pairs and chains. This is alpha hemolytic as well. So we have a greenish, look at that. See, this is what alpha hemolysis looks like. It's greenish around it. It does have some clearing, but it's a greenish clearing. If it was a proper clear clearing, then that would be the beta hemolysis. 50% of healthy people have Streptococcus pneumoniae as part of their normal biota in their respiratory tract. So this is Streptococcus pneumoniae, the guy that we're basically associating with serious pneumonia and with serious meningitis. And so it's normal to have this in your biota, but it should only be there in small numbers, and it's usually going to be in the upper respiratory tract. Moving into the lower part is when the group comes to pneumonia. So you inhale this into the deeper lung tissues, and then it will start to thrive there and lead to the development of fluid in the lungs. Old age, season, underlying viral disease, things like that can make you more likely to develop pneumonia. It does have a polysaccharide capsule that prevents phagocytosis and blocks complement action. There is a vaccine available for the pneumococcus. Remember, we talked about that in the meningitis stuff that can also prevent the pneumonia version of this. It is treatable with antibiotics, but it is pretty resistant to quite a few of the antibiotics available. Next is Mycoplasma pneumoniae. Mycoplasma are bacteria that lack cell walls. So these guys, their shape is called pleomorphic or irregularly shaped because they don't have a cell wall to help with the shape of these guys. And this leads to the presentation of atypical pneumonia. When we talk about pneumococcal pneumonia, this is the kind of pneumonia that makes it really hard to breathe. You're gonna have low oxygenation. You're probably gonna be bedridden from it. But with Mycoplasma pneumoniae, this bacteria will cause walking pneumonia. So you'll actually have inflammation and fluid buildup in the lungs, but you're able to still function and carry out your daily things. I'm not saying you won't feel sick, but you won't be bedridden by this guy, and that's walking pneumonia. So anytime you've heard about somebody having walking pneumonia, this is the cause. Then we have Legionella pneumophila. This causes Legionnaire's disease. Legionnaire's disease is whenever we have basically tap water, cooling towers, spas, ponds, and fresh water that have become contaminated with this bacteria, Legionella pneumophila, and then the steam or the aerated moisture from these guys, you can breathe that in and the bacteria get in. It's not super common, but it was discovered at an American Legion convention, which is basically like veterans. And they had like a convention that all these old guys got together, and they the building, the cooling towers of the building had this bacteria in it, and these guys got pneumonia after that. So yeah, we call it kind of, it's one of the versions of sick building syndrome. There's this, and then we have like black mold and things like that we can kind of categorize into sick building syndrome. But where like the building, some aspect of it is contaminated and it can lead to people getting sick from being in the building. All right, next we have Histoplasma capsulatum. This is not a super common one. We see this, we can see Histoplasma capsulatum associated with like elderly people and people that have lowered immune systems. We call this histoplasmosis. And so this is basically an infection that's associated. We also call it spelunker's disease. So you may have heard about this like bat droppings. So this is the infection that you can get from being exposed to bat droppings because bats have this in their feces, Histoplasma capsulatum. Signs and symptoms can become similar to tuberculosis in chronic versions of this. And most serious forms are going to be found in people that have HIV, but I've seen, just elderly people have had histoplasmosis before. All right, pneumocystis gyrovecii. This is a fungal disease that is normally associated with people with AIDS. Most people aren't going to be getting pneumonia caused by pneumocystis gyrovecii unless you have HIV. So it's one of those kind of defining illnesses. Oh, you have pneumonia. Oh, it's caused by a fungus, pneumocystis gyrovecii. Then we're going to be looking in to see if you have AIDS because it's a very common indicator of that. All right, healthcare associated pneumonias. About 1% of people who are hospitalized will develop pneumonia. It's usually associated with ventilation equipment, and as well as endotracheal and tracheostomy tubes and ventilators in general. If you are part of the 1% that gets pneumonia in a hospital, you have a 30 to 50% mortality rate, which is awful. Yeah, that is awful. And so this is usually going to be associated with the fact that people who are in the hospital and get these respiratory infections are bedridden already. So they're not able to ambulate, they're not able to pump their lymph naturally. And so there's that issue, but they're also usually like laying down and they're stuck that way. So a lot of these people are going to be associated with people that are already like unconscious and not in a active, awake state. And so they aren't able to sit up and cough and things like that. Causative agents of this are going to be things like MRSA, Klebsiella pneumoniae, and Enterobacter. These are bacteria that are just kind of everywhere all over the place. So these are not usually associated with pneumonia, but when you get healthcare associated pneumonia, this is an HAI that is associated with bacteria that are in that environment, causing illness in people who are already in a weakened state, leading to that pneumonia as a result of it getting into the respiratory tract, usually associated with the respiratory tract equipment. So how do we prevent it? This is usually from aspirating bacteria and fluids from the upper respiratory tract. So elevating a patient's head can help with it. Practicing deep breathing and frequent coughing if the patient is conscious and are able to do that. And then, of course, proper care of the ventilation equipment. This is easier said than done if you know anything about operating in a hospital, that you can do everything that you think that you can to help some of these patients. If they have a decreased immunity because of just the stress of whatever trauma they might be there for, usually some other illness brought them there, that they're going to be more susceptible to infections. So we have people that are using like sterile, tubing and things like this. And then like 2 days later, they will look at the aspiration of fluid from these patients that'll be like thick and good green and stuff like this. And what can you do to prevent that sort of stuff? And it's a lot harder than you might think. So 30 to 50%, and this is including being aware of it and treating it. All right, hantavirus pulmonary syndrome. It's caused by a virus called hantavirus. It can lead to the development of hemorrhagic fever. It's less common, but in hantavirus pulmonary syndrome, this is something that develops as a result of that virus moving in and really wreaking havoc in the respiratory system. This is basically transmitted by rodents. So rodents pee and poop in your house, and then you might go and clean up in your house and sweep and things like that, and then you'll breathe in the dust, and then that leads to exposure of that. This is mostly associated in the Southwest United States, in the Four Corners area, and we discovered the first cases of this in the 90s. So it's a pretty new disease, it's an emerging disease, and it has a mortality rate of 33%, which is pretty high. Again, it's not super common, but you often will get fever, chills, feeling like you're sick. Okay, right. That's what these symptoms are. I think we can agree with this. The prodrome phase, you'll feel like you're sick. You have like a respiratory disease and maybe some GI symptoms. Cough is pretty common, but it's not always predominant. But then edema will happen in the lungs over time due to the presence of the virus. And then that can lead to issues with getting oxygenated, having severe breathing difficulties, and you can die from that. Again, it's airborne via dust contaminated with urine, feces, or saliva from infected rodents. It's more commonly harbored by deer mice. We have increasing incidence in the United States, west of the Mississippi River. And so people have started having increased issues with this in their homes. If you have mice in your homes and you're at risk for this. Again, it's not super common, but that doesn't mean it's not become more common. But yeah, here's some places where we've seen it. All right, and that is it for lecture today. I'm not going to go over the questions because we're done. So please take the time to go over the questions yourself and make sure they make sense. If they don't, then email me. We're going to head up to the lab for our review since there's another class after ours. We'll head up there and do our lab review, and I'll help share some note-taking tips for you guys as well.